The first deep learning assignment that I did was, the first deep learning assignment that I did was talking about how the Pfizer vaccine reacts to beta coronavirus alpha variant. So yeah, it's very nice. So based on the article, this is the first half of the article talking about alpha coronavirus. In specifically its spike proteins. And I used this to link with another article that I don't have anymore. Very sad. Yeah, because it got canceled. But it was talking about the Pfizer vaccine and how it attacks the spike proteins, or not attack, but create antibody to be against the virus. So the vaccine contains an enzyme called TMPRSS2, which is able to repel the virus or maintain it at bay and slowly dissolve it until it dies. Yeah. Yeah. Very simple. Very nice. Very simple. Yeah, I made it extra simple, bro.
That's all with the first one?
Yeah, for the first one. The second one is, let's see here. The second one was talking about molnupiravir, which is a new pro drug that people want to use or is in clinical trials to go against SARS-CoV-2. So what it does is that it causes mutagenesis. So that is to mutate the SARS-CoV-2, like put a stop codon so that it cannot be like a COVID virus anymore. It cannot reproduce, cannot do this and that. Well, in terms of what it actually does is, yeah, it just puts a stop codon. It prevents the virus from mutating. Yeah. So I'm gonna compare it to remdesivir. You know remdesivir? So that's the RNA-dependent RNA polymerase, right? It's able to, remdesivir is able to disguise itself into the codon, right? So when the RNA of the virus tries to replicate, molnupiravir won't be detected, but remdesivir is detected, therefore unaffected. So remdesivir would be kicked out. Molnupiravir is more of like, it has a disguise, bro. It actually disguises itself and passes undetected, leading to a mutated replica, stop codon, or mutation upon, not able to do. And then molnupiravir within the article, it talked about how it was against mammalian cells, so like mouse, for example, and they state that there's possible long-term side effects, but unsure. So they need further investigations. Yeah. My last one, journal critique. Not that. Journal critique was of the article about neuroinvasion of SARS-CoV-2. So it's basically, the COVID-19 attacking the brain. Yeah, causing irregularities, like patients who have already dealt with COVID and survived. They say that they have memory loss, headaches, right? That's what this article addresses a little bit, but it uses 3 ways, and that is, well, firstly, it cannot get I can't take your brain, cut it open and see if there's COVID inside, right? So instead they make human organoids to test the brains. So petri dish cell brains. They use mice brains, in total 35 replicas to test. And then they also had 3 postmortems that they saw that there was neuroinvasion. I mean, when I was reading this, it wasn't like they're finding that, oh, there is neuro invasion. It's more of like they already know that there's neuro invasion, but they want to find out how it goes into the brain, because there is like a blood barrier that blocks your neck and your brain, so like viruses can't really go through. So how does it go through? And they did identify a little bit about it, but they were still unsure. They stated that maybe it went through the nasal cavity and went up because it's very close, or they went through the blood barrier. So that's that. This one? Sure.